Dear ladies and gentlemen, dear Chairman, so it's a pleasure for me to present our results on biomarkers in PRRT patients with personalized high activity. So the motivation to use personalized treatment is that we can define the absorbed doses in critical organs and tumors before the therapy. So we do pre-therapeutic dosimetry and then use patient-specific administered activities. And what we did in our studies, we want to have a better assessment of the radiation damage. So we combined internal dosimetry and biodosimetry using the biomarkers gamma H2X and 53BP1. And then we described the dose and time dependency of those markers in blood lymphocytes in the PRRT patients with the personalized high activity and compared it to previous studies that used the standard treatment activity. So shortly after double-strand break forms, the histone gamma H2X is phosphorylated, and then it is called gamma H2X. And furthermore, 53BP1 accumulates in the close vicinity to the double-strand breaks. And then we have two biomarkers that can be used to assess the DNA double-strand breaks. To visualize those markers, we used immunofluorescence staining of the lymphocytes. And then you see in the nucleus, which is seen in blue, these little foci. And we used co-localized foci, so only if there was a gamma H2X and 53BP1 foci visible in the same spot, we said this is a DNA double-strand break. So the DNA damage assay is used as a quantitative proof of radiation, and it is assumed that one focus represents one double-strand break. And it was shown after external irradiation that there is a linearity between the number of radiation-induced foci and the absorbed dose. However, for internal exposure, for example, which we have in nuclear medicine, there is not a lot of data available. And the difference between the external irradiation is that we have an internal irradiation and continuous irradiation with a decreasing dose rate. So for introduction, for the dosimetry we did, I just want to give you a short overview how we assess the blood-based dosimetry. So for the lutetium, we have to use two compartments. So first, we have to consider the self-irradiation of the blood, so blood as a source organ, and therefore we only take the beta radiation that originates from the activity in the blood into account. And then we have the second compartment, which is the whole body, and the gamma radiation irradiates the blood from the whole body. So to calculate the absorbed dose to the blood, we need to know, of course, the administered activity. We need to know the S values, which is the mean absorbed dose per nucleon disintegration in the target organ. For the blood self-irradiation, we need to know the time integrated activity coefficients for blood and also for the whole body. And the time integrated activity coefficient is nothing else than the area under the curve of our time activity curves from blood and whole body. So to get the time integrated activity coefficients, we have to collect blood samples at different time points and also do whole body measurements and then quantify the activity in the blood and in the whole body. So for the activity in the blood, we performed well counter measurements, and for the whole body, we did whole body scans at 1 hour and 24 hours, and also performed external dose rate measurements just above the patient bed for 5 time points. For the pre-therapeutic dosimetry, we administered about 200 megabecquerel to the patient and also performed blood sampling and camera measurements and also one SPECT-CT at different time points. And the limit was the absorbed dose to the kidney, because that is the critical organ in the therapy with 23 Gray. And according to the local restrictions in Germany, or especially maybe also in Bavaria or in our department, the limit was 20 gigabecquerel because we're not allowed to handle more activity in the lab. And so here you see the patient data. So we only had 3 patients for this personalized treatment with high activities, and the age of the patient was between 60 and 76. And the activities given to the patients, the lowest activity was 14 0.4 gigabecquerel, and the highest activity was 19.3 gigabecquerel. And as you see here, this is the absorbed dose to the blood after 48 hours, and on the right side, the total absorbed dose. And you see that not the patient with the highest activity also received the highest absorbed dose to the blood. So the patient with a low activity had 196 millicray, and the patient with nearly 19 gigabecquerel had only 128 millicray. So it's not only a matter of the administered activity, what the absorbed dose is to the blood. And for our analysis of the biomarkers, we need to take blood samples. So the first sample is just taken before the therapy to get a background value for the foci. And then we took blood samples at the first day every hour, up to 4 hours, and then 24 hours, 48 hours, and 96 hours. And to assess the radiation-induced foci, we have the total foci number minus the background foci number. And to separate the lymphocytes, we use a special method. So we take the blood samples and use special centrifugation tubes. And here you see the whole blood above a gel barrier. And after centrifugation, you see the separated blood. On top is the plasma, and here is our lymphocytes, which we can easily take off and then fixate. And afterwards, immunofluorescence staining can be formed, which was done at our partner institute in Munich, in the Institute of Radiobiology. And then the foci were manually counted, and as I said, we only calculated counted the co-localized gamma H2X and 53BP1 foci. So here you see a study which we performed just before the patient studies. It's an in vitro calibration where we added iodine and also lutetium to blood samples, which we took from volunteers, and added different amounts of activity and performed a Monte Carlo simulation to get the absorbed doses to the blood. The samples were incubated for 1 hour, and here you see that we get a linear function of the average radiation-induced foci with the absorbed dose, as it also was shown for the external irradiation. And we have about 0.0145 radiation-induced foci per cell in millicray. And in this graph, you see the absorbed dose to the blood as a function of the time of the administration, and the solid line, and the dotted line is the dose rate of the different patients. And so as you see, we have a really high dose rate at the beginning of the therapy, and then the dose rate gets smaller and smaller. And so here you see that the patient with the highest activity has also the highest absorbed dose, but as I said, the patient with the second highest activity has the lowest absorbed dose to the blood. And this graph gives you the radiation-induced foci as a function of the time after administration. So within the first couple of hours, you see a very steep increase of the radiation-induced foci, and then you see a decrease of the radiation-induced foci, and that comes probably because of the high increase of the absorbed dose to the blood at the beginning, and then because of the decreasing dose rate, we have competing processes of foci production and disappearance of the foci. That's why we have this decrease in the foci. However, with the patient with the high activities and also the high absorbed dose has a quite high amount of radiation-induced foci even after 96 hours, whereas the other patients drop at later time points. And if we now look on the radiation-induced foci as a function of the absorbed dose, which was already shown in the morning highlight section by Professor Hoffman, we see that we have a linear function of the radiation-induced foci as a function of the absorbed dose to the blood within the first 4 hours. And you see the red dotted line is our in vitro calibration curve, and the black solid line and the orange dotted line, which is not really visible because they just lie above each other, are the in vivo calibration curves of the high activity patients and the normal activity patients. So normal means the standard activity of about 7 gigabecquerel. But after this time, we see again the drop of the radiation-induced foci, and we don't have you can't tell a function of the absorbed dose. So it's only possible within the first hours. And the slope of the functions of high activity patients, normal activity patients, are pretty the same. But what is a little bit, or what we found out, that if we just look at the 4-hour time point of the radiation-induced foci, we see that we had about 0.12 radiation-induced foci per milligray at 4 hours. And it's the same as the slope. But if we look on the standard activity patient, it's a little bit busy, this slide, but for the normal activity patients, we had much more patients. So the variety of the foci numbers is a little bit bigger than for the high activity patients. But if we also perform the in vivo calibration for the normal patients, we get the similar slope, as I said. But if we look at the 4-hour time point, we have a much higher number of radiation-induced foci as normalized to the absorbed dose after 4 hours. And that might be a hint that after high activities, the repair rate is a little bit higher than for the normal activities. And the absorbed dose to the blood for the normal activity patients is about 80 milligray compared to the about 200 milligray in the high activity patients. So in summary, we can say it was possible to also analyze the biomarkers in patients during the first personalized peptide receptor radionuclide therapy, and the slopes within the first 4 hours of the personalized and the standard treatment are pretty similar. But if we just look at the 4-hour time points, we see differences. So for the normal standard activities, the radiation-induced foci values normalized to the blood dose are much higher than for the personalized high-activity patients, and this could indicate a faster damage repair using higher activities. But in total, the findings align well with the previous results. And with this, I would like to thank you for your attention.
Thank you very much for this very interesting talk. Anyone have any questions on that?
I have a quick one before somebody gets up. This is with Luteef and the beta emitter. Are you going to be able to do this with the alpha emitters as well?
Actually, we are doing some studies on that, but we're pretty at the beginning, so it's difficult to tell anything right now.
Your data are very similar to what we found using gamma H2X in terms of the rapidity of the upstroke, if you like, of gamma H2X. The thing that I won't pretend to understand is the high variability, what happens after that. Out at 4 hours, 24 hours, 48 hours, some patients have persisting gamma H2X in their blood and others have virtually none. And it seems to vary depending on if it's their first cycle or their second or their third. And we often find ongoing gamma H2X foci in patients who've had treatments some weeks ago. Do you have a handle on that? Do you have any ideas of what it might mean?
For this study, we just had the patients that came for their first treatment. So we didn't look on patients with the second, third, fourth cycle. But we also saw with a normal study with a 7 gigabecquerel, we also saw this variance. So, but at the moment, we don't have any clue why some have the persisting foci and some don't. Even if they have really low absorbed doses, they could have persisting foci, especially for the personalized treatment. Can I go back somehow or no? The patient with the lowest absorbed dose had the highest number of foci, or really high number of foci as well after 96 hours. So I think it's not really, we can't correlate it to the absorbed dose, so there must be some other factors.
I mean, obviously we're all worried about inducing second malignancies, particularly myelodysplastic syndrome and leukemia. do you think that the rapidity of DNA repair in your lymphocytes or circulating blood cells might be a biomarker of the risk of myelodysplastic syndrome occurring in these patients?
We hope so, but I mean, we also looked a little bit on the toxicity data, but we couldn't relate it to the absorbase either, and also not exactly to the high number of foci. So we hope we could correlate it somehow, but I think we're at the moment on a really early stage, at least at our studies. But we really hope that if we look on studies where we have higher toxicity in the system, we might have a better marker to assess this.
David Dick, University of Iowa. So my question is actually right along those lines. You stated the assumption earlier that you're saying that one foci represents one double-strand break, and then the question is that a valid assumption to make, or are we seeing multiple double-strand breaks in that one foci? And of course, at higher exposure rates, you could expect that maybe your foci end up, you've got multiple events that get so close together that you just see one foci, and therefore that would give you, maybe explain some of the things that you're seeing. So have you done any work to look and actually measure the amount of double-strand breaks that you're seeing when you do this? There's a number of markers out there that you can look at for DNA fragmentation.
I mean, what is obvious that if as you said, if they're too close together, we won't be able to see the difference of the foci. But right now, we are not able to distinguish if it's only one or two foci. But we have really low doses, so I think that's more a matter of fact if we have quite high doses above like 2 Cray or something in the blood, where you really have the occasions next to each other, or with alpha irradiation. So then we need to use another measure how to assess the foci. But it's true, it's difficult to, but we haven't done any studies on that yet.
I can tell you from looking at the immunohistochemistry that you often see a track, like a cloud chamber, through the nucleus of several in a row. So I think that most of them, you're probably single double-strand DNA breaks that you're seeing. But it's possible that by stochastic reasons you'll get some that pile up on each other. But I think most of them are probably single from having looked at quite a few of these.